short lecture is going to just give you a brief review of mechanisms of transport that are going to be very important in understanding the processes of ultrafiltration in the renal tubule. So before we even get into the specifics of things like the parts of the renal tubule, the different antiporters and symporters, and what part of the tubule is soluble for what, and then understanding solute movement, we have to first just briefly review the different mechanisms of transport, because that is going to be really important as you start to tie these all together to create a story of movement throughout the tubule. So glomerular filtration, hey, come back, buddy. There you go. Don't run away. The glomerulus, remember, is just filled with fenestrations, and anything that can get through those holes enters Bowman's capsule. That doesn't mean that it's going to be destined for the urine, because some of those processes or some of those things lost to the filtrate are just small, and we might want to retain them. So there's going to be a different kind of alterations made to the filtrate so that we are losing a nice concentrated urine filled with waste products, but then retaining in the bloodstream anything that was initially lost to the tubule just through simple glomerular filtration, and that's called ultrafiltration. So this is going to involve a huge variety of transporters and a general understanding of where they're located, and then the functionality, and just a review of the way that things move throughout cells. So in this section, we will be discussing how all of these processes go together to create the most concentrated urine. So a concentrated urine, urinary excretion, is glomerular filtration, again, anything that leaves the glomerulus, minus anything reabsorbed back into the bloodstream through the ultrafiltration process, and plus any extra components that are added to the filtrate later on in the secretion process. So when we're talking about movement throughout the renal tubule, so here we have a section of the kidneys. So in yellow we have the cortex, and the corticomedullary border is this area right here, and then we have the medulla. And what you'll see is initially everything small is going to make it into Bowman's capsule and then start to traverse through these different parts. We have first the proximal tubule, which means the closest part of the tubule to the glomerulus. There is the loop of Henle, is the eponymous name. It's also called the renal, like the glomerular loop or the nephron loop, but loop of Henle, which has different segments on it. There's a thicker section, there's a descending section, there's an ascending section that's thin, there's an ascending that's thick, and then there's the site of physiologic regulation, the distal tubule, which means it's far away from the glomerulus, and this is going to be the site of a lot of secretion, and it's also going to be the site where we can have the activity of things like aldosterone, so some hormones working. And then all of these things through this process, as we're adding different things, as things are being traded back and forth between the blood and the urine collection, we're finally going to deliver it all to this collecting duct, which then is going to move towards the renal pelvis. And the collecting duct is also a place where hormones can act, primarily ADH. And we'll take a look at that a little later on. What you're seeing here, this is a nice slide to start with. Not only do we have different objects and the way that they're moving, we also have a classification of the transport systems. So there are going to be processes of active transport, meaning that they require energy. There's going to be passive transport, which typically is just high to low concentration, not requiring any extra energy, just using kinetic energy. And then there's secretion, which is going to mainly involve active transport processes. So there's a lot of different activity going on in the tubule, and you are going to have to know all of it, unfortunately. So this is a good place to start with everything labeled. But first, again, we're going to go through just these processes. So we've talked about glomerular filtration. We know it's non-specific. It's just governed by the size of whatever the substance is relative to the size of the fenestrations and slit pores in the glomerular capillaries. So this filtration is determined by the rate or the pressure of fluid in the glomerulus. We call that the glomerular filtration rate, and it's also affected by the plasma concentration of that substance. So we're assuming that things are free and not bound to plasma proteins, because we do know that drugs and hormones, they can bind to things like albumin, and albumin shouldn't be filtered, right? So once we get to the tubule, we are going to go through this more specific reabsorptive process, which is highly selective and is going to allow the body to create the ultimately most concentrated, accurate urine. And you can start to look at some different products. As we've mentioned, glucose is something that is filtered, and it should be completely reabsorbed and not excreted. 100% of glucose should be reabsorbed back into the bloodstream. If you do have a patient with glucose in the urine, it usually means they have low insulin, and that's a sign of diabetes. So low insulin, because insulin moves glucose. So glucose shouldn't be present in a typical urine. Bicarbonate is something that is filtered and, again, almost completely reabsorbed. So 4320 millicues a day is filtered by the glomerulus, and almost all of it is reabsorbed back into the bloodstream. Small amounts are excreted depending upon the pH of the body. Most of it's reabsorbed because we have to balance the acid-base balance in the body. Sodium, same thing. We have a lot that is filtered by the glomerulus. A large amount is reabsorbed. But there is going to be a larger amount than other products that will be excreted. But usually we're reabsorbing most of the sodium as well, because where sodium goes, water follows, and we want to keep our water in the plasma, in the bloodstream. Sodium travels with chloride. Sodium is positive, chloride is negative. They can be co-transported together, and chloride just passively follows sodium, kind of like the tail on a dog. So it is very similar rates where the amount filtered is very similar to the amount reabsorbed. There is a little bit excreted, but pretty much all, like greater than 99% of chloride is reabsorbed. And then we're starting to get into things that we have a little bit more of. So potassium, there's 756 millicues per day filtered, 664 reabsorbed, 92% excreted, only about 88% of potassium is back into the bloodstream. Then we have our waste products. So urea is going to be the place where the nitrogenous wastes are going to be housed appropriately. And if we don't excrete urea, that leads to a process called uremia. So high levels of uremia, which is an unhealthy state. It causes neuropathy and other types of damage to the body, even like pericarditis, pleuritis. So it's important that the amount that's filtered, not all of it should be reabsorbed. There should be a good amount of excretion. So it should be pretty well matched with reabsorption and excretion. So it's about 50-50. 50% urea is reabsorbed back into the bloodstream, whereas 50% every day should be excreted. And then we have our nice important marker of creatinine in grams per day. Remember, creatinine shouldn't have any action in the body. So if we're making 1.8 grams of creatinine a day, there's none that's reabsorbed because we don't have any use for it in the body. So all the creatinine that's created should be excreted and cleared, none reabsorbed. This is a really good marker for telling you what the GFR is and how efficient the kidneys are at excreting products. If you do see creatinine at high levels in the urine, that tells you that the kidneys are failing, and that's when you start to investigate things such as dialysis protocols. So just in understanding ultrafiltration, we're losing and adding things from the filtrate so that the urine ideally contains a high amount of waste products and a low amount of materials that are meant to be components of the bloodstream. And this process of reabsorption is going to involve both active and passive transport. So if we look at a very close-up view of what we are talking about in this system, whoops, come back, buddy. We have, this area would be I'm going to make it yellow because this is the lumen, or the inside of the tubule. So the lumen of the tubule. Then we have the cells of the tubule itself, tubular cells. They're tubular, they're the epithelial cells that make up the tubule. And then we have the capillaries, the peritubular around the capillaries, like a net wrapping around the different parts of the renal tubule so that things can be transferred back and forth depending on what is needed and what needs to be excreted. So we're going to begin with just the simple filtration at the level of the glomerulus, and now we just have a whole bunch of products in the lumen of the filtrate. And that filtrate is going to have to move either between cells, we call that the paracellular path, so if things can fit through there, they can just move through bulk flow, high to low concentration. That's pretty simple. Then we have a transcellular path. Trans means across, so that means things can move across the cell out into first the space and then reabsorb back into the bloodstream. And then different solutes, because things are gated based on their charge. So we don't want to have free movement of positive and negatively charged ions into cells, because that's going to change their resting membrane potential. So we will have things that are charged moving through different processes that can be an active type of transport, which means there's energy. So it might be in the form of movement through a pump run by an ATPase. There's cases where we'll just have passive diffusion, which means it's going high to low concentration using kinetic energy. And then water, because water is special when it dissolves over a membrane. So the diffusion of water over a membrane is called osmosis. Osmosis is something that can happen in the paracellular or transcellular path. And remember, solutes move from high to low concentration, whereas water is going to move to solute concentration. So it moves to places of high solute concentration. Okay, the Starling forces are going to really govern these processes also. Remember, the Starling forces involve the Donin effect, which states that proteins, sorry, my writing's really messy, so proteins are negatively charged and the contents of the plasma are mainly positively charged. So protein and plasma fluids are attracted to one another. And then we have hydrostatic pressures and oncotic pressures associated with the colloid. So these attractive forces plus the pressure governed by the amount of fluid is all going to come into play. Very important. So just a little review. We have passive transport here in the simplest sense using kinetic energy. We have simple diffusion, which is when solutes, again, they move from high to low concentration using kinetic energy. If those things are lipid soluble, or gases do this really easily, they can just diffuse right through through the membrane, and we don't need anything to move it there. There's also facilitated diffusion, which is still passive because there's no energy. So this is in the case that there is not a solubility of these various solutes, so therefore they take just a doorway into the cell. So these are just channels. So that can either be through a receptor pore that opens, or just through a mediation where they bind and then the cell changes shape to move it in. So this would be like ghosts can move right through the wall because they're ghosts. They can dissolve through a wall. People can't do that. We have to take doors. We have to move through an open door. This would be like a mechanical rotating door. So these are still high to low concentration, and they don't use energy, so they're all passive. Active transport literally means that there's energy required, and you hopefully know by now, wherever you are in the world, if you're watching this video, that the most common type of ATPases are sodium-potassium pump, which just means that it runs on ATP. That's its energy generator. In the same way electricity is what generates power for the appliances in your home, ATP generates power for different cellular just proteins. This is an enzyme. So when things have to move against their concentration gradient, so remember things flow really easily high to low concentration. If we want to move them in the opposite direction, or if we want to have a tighter control over that movement, we have to use some form of energy. So here we would be trading things like 3 different products, maybe 3 sodiums for 2 potassiums, and that's a really common type of active transport. It would just use ATP as energy. So this active transport is required anytime we're moving against the concentration gradient. So you should hopefully know sodium and chloride are really high outside of cells and potassium's high inside of cells. So it's really easy for potassium to naturally flow outward. But we don't want anything happening that allows free movement of things like sodium and potassium, because that will change the charge of this membrane. But even if this breaks down, so that does happen, we talk about it in cellular injury, and this happens in the kidneys actually. If there is a breakdown of blood flow to the kidneys and there's no oxygen, then we can't generate ATP. That means these pumps are going to break down. If sodium finds a way to get into the cell, it will rush in, and where sodium goes, water follows. So that cell will swell, and if we don't restore blood flow and restore ATP, then that cell will eventually burst and become damaged. So we need to tightly control this. So active just means it requires energy. So this is our most common ATPase in the whole body. So we've got our potassium really high inside of cells, our sodium really high outside of cells. That's the natural tendency of things to, or way for things to move. So the sodium-potassium pump uses ATP to trade sodium for potassium. So again, sodium would naturally flow in, but this is to do things like move sodium out because it's against the concentration gradient. So sodium and potassium are traded for one another. This is the most common ATPase in the whole body. We're trading 2 sodiums for 2 potassiums. And this just uses the phosphorylation of ATP into ADP to drive a conformational change and then to traffic things in a direction. Then the other type of active transport, instead of using ATP as a power source, it uses the indirect energy, kinetic energy associated with the movement of another ion. So for example, this is co-transport of sodium and glucose, whereby these 2 processes happen because like sodium we know is really high outside of cells and really low inside of cells. So there's a high amount of kinetic energy associated with the movement of sodium. Glucose can piggyback on sodium and utilize its kinetic energy as its energy source for movement. So you'll see co-transport with sodium and glucose, but then also insulin and glucose, for example. So it's still energy, it's just not ATP derived. In this case, it's using somebody else's energy. So it'd be like literally climbing on somebody's back and getting a piggyback ride up the stairs. You're still moving, you're borrowing their energy, and you're just along for the ride. Okay, so these types of processes happen all over the cell, and you also have to remember that the cells themselves still have their own natural resting membrane potential. So it's going to become a little bit more complicated as things like sodium move from the lumen into the bloodstream. So if we follow sodium here in the lumen, some of it's going to move through this paracellular pathway and just in a bulk flow right into the blood. But we'll still have some active transport with sodium and potassium because this is still a cell, and this tubular cell needs to maintain its resting membrane potential of negative 70 millivolts. So it becomes more complicated because not only are we going to be moving substrates, but we also have to maintain the resting gradients for the physiologic cells in the body. So in this picture, you're seeing sodium, again, moving in a paracellular way, just into the bloodstream through bulk flow, but then it can also move in a transcellular way and be traded, pumped out, so traded for potassium. So in this case, we'd have active pumping of sodium out, traded for potassium, because potassium should be high in cells. And then there's still another ATPase over here trading sodium for potassium. You're going to find sodium-potassium pumps all over the body. They're the most common types of ATPases. They exist on every cell, really, that has physiologic function. So these are going to maintain the functionality of the tubular cells. Okay, so when you are thinking about the way that things move and where these pumps are located, you can take for granted that there's sodium, potassium, ATPases in every cell, and sodium travels really well through the paracellular pathway, but it usually takes the transcellular route because of the attraction that it has naturally for this low solute level of sodium inside of cells. So there's a bunch of sodium, potassium, ATPases in cells, moving sodium out, moving potassium in. Those are normal concentration gradients that helps maintain that cell's functionality. So this is going to create its own concentration gradient. Keep in mind these are running all of the time, and this is going to keep this cell running, but it's also going to mean that we have the perfect instance to create the movement of sodium in that it will move high to low concentration. And also positive and negative ions are attracted to one another. You'll see in a lot of tubular cells active transport of glucose. There's specialized glucose receptors. Some are on the luminal side, some are on the other interstitial side, the brush border side. These active transporters in the tubule are the S-GLUT1 and 2. They co-transport glucose and sodium together. And then on the apical side, we have the other glucose receptors, these GLUT1s and GLUT2s. So we'll see active transport using the energy of or borrowed from sodium to bring glucose into the cell. So that's a co-transport process. And then we have just a GLUT transporter on the other side. So this is called a symporter. Remember, symporters are not receptors, geez, sorry, I'm getting tired. They are proteins, but that move two substances in the same direction, symporters. Then there is also an antiporter. This is a counter-transport system. So here we had co-transport, glucose plus sodium. There's also antiporters, which trade hydrogen ion for sodium down here. Sodium-potassium pumps and ATPases are also examples of antiporters. So again, take for granted that all cells in the body should have sodium-potassium pumps to maintain the normal resting state. And then we're going to start to see symporters, antiporters, co-transporters, all types of cells. So glucose can be moved most prominently through secondary active transport. How do proteins move? Proteins, you might remember, move through pinocytosis, which is associated with the movement of liquids. So proteins, here in red, they bind to their receptors, which are in an invagination in the cell. Those are called clathrin-coated pits. And when those receptors are all filled, which means they're all saturated, there's actin and myosin in these cells. So this actin and myosin, it snaps the vesicle shut so that now we have a vesicle filled with a protein that can be brought into the cell and then utilized. So they attach to the brush border side. We have invagination. That's called an omega structure because it looks like the letter omega. And then the vesicle brings things in that can be digested. This is the way that we move things like LDL, move proteins, so that reabsorption is through pinocytosis. It can't just dissolve through the membrane because proteins are really big. They have to be brought in their own specialized vesicle. There's actin and myosin here to snap those shut. Actin and myosin requires ATP to do this process, and then there's also a kinesin molecule associated with vesicles that walks the vesicle along, which also runs on ATP. So this is a type of active transport. There's a lot of energy involved in pinocytosis, from the creation of the vesicle to the transport of it. Okay, and then there is a point where the enzymes that bring things in directions, they just get maxed out. That's called their saturation level. So it's called Kmax is the maximum amount of workload that an enzyme can do. It's the maximal rate of transport, and it's just due to saturation. That would be like, there's some point in time, like if you were at the grocery store right now, if there's 100 people in front of you in line, there's only so fast that person who's checking everybody out can work. So that's the saturation point. So this transport maximum is just associated with the saturation of transport systems. So when you look at different things, they all have their own saturation point. That's usually when there is a bottoming out or flattening out of the curve. Here what you're looking at is glucose that's filtered and then reabsorbed or excreted. So we know all glucose is filtered, that's just a standard constant process because it's just moving from the glomerulus into Bowman's capsule. And then if we want to bring it back into the bloodstream, because we should, right? If we are going to reabsorb it, it has to come in through those co-transport systems, and they can only work so quickly. So this is the maximal level that we can work at. It's not as fast as simple glomerular filtration. The excretion process, so that is associated with the plasma concentration. If there's a huge amount of glucose concentration in the plasma, then excretion will be driven higher. When you look at somebody with diabetes mellitus and there's a huge amount of glucose in their urine, this is really uncontrollable. But just for the bottom line, all enzymes, they can only work so quickly. So even though we can filter everything really fast out of the glomerulus, once we start physically reabsorbing things back into the bloodstream, it has to be done by these transport systems, which can only go so quickly. So these are the transport maximums of different substances, so how quickly they can be moved in amount per minute. These are all products that are actively reabsorbed, so glucose, phosphates, sulfates, amino acids, urates, lactates, plasma proteins. And then there's substances that are actively secreted, which means they're squirted into the tubule to be removed. So this is, you don't have to know the numbers for me, but things like your boards might ask, like which has the highest or lowest transport maximum out of substances. Okay, and then just keep in mind that if we are moving solutes, that's also going to drive the movement of water. When things move water or have the ability to move water, they're called osmoles. Some things have high osmol activity. Sodium is one of the most effective osmoles. So taking a look at this picture over here, we've already said when you are memorizing which receptors and cotransporters, symporters, antiporters in each area, you should keep in mind that all cells have sodium, potassium pumps. Those are antiporters. Okay, so this is just using two example substances. So here we have substance X, which was moved from Bowman's capsule. It's being reabsorbed now into the bloodstream, so we have substance X over here. Now we have substance Y, which is being co-transported with sodium, so Y is going to move out here into the plasma. What you will see is that when things like sodium, which is moving out of cells through this active process, this active transport, when sodium gets really high, that's also going to drive the concentration of water, because where sodium goes, water follows. But also just the idea that X and Y, it doesn't matter what they are solutes, and water will move to areas of high solute concentration. The final thing is just urea movement. So these processes are driving the movement of water because water moves high to low concentration. Urea is something that is passively reabsorbed because it is in really high concentration here in the tubular fluid, and it will move down its concentration gradient to be reabsorbed. Remember, about 50% of filtered urea is going to be reabsorbed. And then just tying this all together, passive diffusion is going to involve not only water movement, because we know water is going to follow high solute concentration. So in here, anytime there's a red arrow, it means it's moving into the bloodstream. So when sodium is moved into the bloodstream, that's going to drive water to move there too, right? So we know that that's an osmolar activity. But also keep in mind things like charges. We know sodium and chloride move together because they have an opposite charge. So you have to not only remember that solutes will move high to low concentration, that water will move to high solute concentration, you finally have to remember that negative and positive ions are physically attracted to one another. So when something positive moves into the bloodstream, that's going to drive chloride to be reabsorbed also, so that chloride will passively go into the bloodstream as well because of the movement of positive sodium. And that's the final lesson of transport. So just remember your transport mechanisms, and then we'll tie that all together in ultrafiltration. All right.